Welcome to the 11th International Conference on Climate Change, ICCC 2025. We are now in Parallel 1, Session 2. In this session, we will watch your Pecha Kucha presentation videos. Let's start with the first presentation, titled A Study on the Potential of Utilizing Mangus Rind as Natural Dyeing.
Hello everyone, I'm Nisa and I'm going to present our study titled A Study on the Potential of Utilizing Mangosteen Rind as Natural Dye. This research explores how mangosteen peel and abundant root waste can be transformed into an eco-friendly dye for sustainable batik production. Behind the beauty of Indonesia's batik lies a major environmental concern. Around 60% of batik waste in Central Java pollutes river. The synthetic dyes, these chemical-based dyes harm ecosystem and human health, urging us to find sustainable, natural alternative for batik dyeing. Indonesia's biodiversity provides many potential sources for natural dyes. One of them is the mangosteen tree, rich in xanthones and anthocyanins. This compound can produce stable, beautiful color while reducing organic waste and promoting local resource utilization. This research matters because it's aligned with global sustainable development goals. By using natural dyes, we reduce chemical pollution, promote responsible production, mitigate climate impact, and protect biodiversity, directly supporting SDG 12, 13, and 15. To address the issue, we conducted an experiment focusing on extraction, dyeing, and fixation process of mangosteen grain. We also tested the color fastness using the international grayscale standard from the Society of Dyers and Colorists to ensure measurable results. We follow four main steps: molding the fabric, extracting the dye, dyeing the cloth, and fixing the color using lime, alum, and Tunjung solution. Each step was tested in the lab to evaluate stability and fade resistance under real washing and rubbing conditions. The results showed that mangosteen green dye achieved fade resistance score of 4 to 5 on the grayscale, meaning very good fastness. All three fixation methods produce stable, attractive color with alum and Tunjung fixation, showing the best durability for daily use. This study demonstrates that mangosteen green dye performed almost as well as synthetic dyes, but without harmful effect. It proves that waste material can be repurposed into high quality, eco-friendly dyes that support both sustainability and artisan safety. Our findings contribute to sustainable textile innovation and circular economy practices. The use of mangosteen green supports responsible consumption, climate action, and environmental protection, making batik not just cultural but also sustainable heritage. Thank you for your attention. This research shows that innovation and tradition can go hand in hand. By reintroducing natural dyeing, we can protect the environment while preserving Indonesian culture's
For the next presentation session, entitled Freshwater Ecosystem Integrity and Climate Smart Management in the Didesa River Basin, Ethiopia, Implications for Resilient Agriculture and Biodiversity Conservation. If you have any questions regarding the previous presentation, please kindly write them in the chat box.
Hello, welcome to my presentation. I am Branu Zaude from Ethiopia, PhD candidate at Jimma University and a lecturer at Wellagga University. My research focuses on linking freshwater ecosystem integrity with climate-smart land management. The problem here is freshwater ecosystems are under synergetic problems of human activities like agriculture, sand mining, settlement, and climate variability. These stresses reduce water quality, destroy habitat, and threaten livelihood. Opportunities we have for the problem is integrating watershed management, habitat restoration, climate-smart farming offers, paths to balance food production with ecosystem health. And also, policy and community participation can drive this transformation. Ecosystem integrity matters here because the Desa River is key Blue Nile tributary sustaining biodiversity and agriculture. Degradation here affects downstream ecosystem and the regional food security, making resilience planning nationally significant. We assessed ecological integrity across 46 sites during dry and wet seasons of the study area, measuring water quality, habitat disturbance, and microinvertebrate communities, and we analyzed by using GLMM model. Collected data of water quality, habitat quality, human disturbance, and microinvertebrates were combined, analyzed, interpreted, and compared with standard guidelines. And also, we have compared downstream and upstream sites. The finding of this research is that water quality and biodiversity varied strongly with human disturbances. Agriculture and sand mining caused higher turbidity and nutrient loads, reducing sensitive taxa of the area. These results identify priority zones for intervention and prove that climate-smart land use can sustain both crop productivity and ecosystem health. They provide evidence for data-driven watershed policy. This finding can contribute to basin-scale conservation planning, freshwater resilience, and regional strategies for sustainable agriculture and biodiversity conservation under climate stress. In summary, this study highlights how maintaining freshwater integrity and promoting climate-smart land management can support both agricultural productivity and biodiversity resilience.
For the next presentation session entitled The Effectiveness of the Instant Spice Basa Genep on Various Traditional Food Products. If you have any questions regarding the previous presentation, please kindly write them in the chat box.
Good morning. I'm Ayu Sodani from Warmadewa University, Bali. I would like to present my research. The title is The Effectiveness of the Instant Spice Basagenap on Various Traditional Food Products. Background in this research, the basic spice Basagenap is a spice used in almost all traditional Balinese cuisine. It consists of 26 spice and spice prepared in a press form, so it takes a long time to prepare the spice. To shorten the spice preparation process, instant Basagenap seasoning is made. Opportunities: enhancing local produce competitiveness, development of product variants, innovation in research and development. Why does it matter? Is research a cultural culinary preparation, practical and efficient cooking, and economic impact and customer awareness? The research object is preserving culinary heritage, providing practical solutions, and supporting local economy. The research process are literature review and experimental design, product testing, sensory evaluation, nutritional analysis, data analysis, and conclusion recommendation. And the found instant basa genep is fresh spice versus instant seasoning basa genep.
Apa? Ano ya? Oh, udah selesai ya? Udah selesai semua. Cepet banget. Kok anunya lo jadi logo ICCC
For the
gitu
next
loh?
presentation session
Udah
entitled
selesai aku
Evaluation
potong
of
aja
Growth
gimana
and
sih?
Physiological Characters of Local Central
Ih,
Java
nadanya
Rice under drought conditions. If you have any questions regarding the previous presentation, please kindly write them in the chat box.
Good morning. We are pleased to present our research title, Evaluation of Growth and Physiological Characters of Local Rice Under Drought Conditions. This research is by Edi Prawanto, Coordinator Fauzia, Muchirahayu, and Pacento from Universitas Maret. I'm Coordinator Fauzia. We will present this presentation. Global warming induced drought-threatened rice, a highly sensitive crop, particularly in Asia, which disrupt its physiological processes. The development of tolerant genotypes through conventional crossing has been very slow, mainly due to a scarcity of donor genotypes and a lack of effective screening methods to identify them. It identified tolerant accession and local varieties are key germplasm sources with desirable traits. While previous research identified the tolerant Powys smalam, this plain study will evaluate Central Japanese varieties to find new sources of drought tolerance. Drought in Asia threatens global food security by impacting rice, the region's primary crop. Water stress causes significant economic losses. Understanding rice's complex physiological and molecular responses to drought is essential for developing new resistant cultivars, protecting agriculture against climate change. Planting was conducted in a greenhouse. Water content of soil was measured in the lab scale and calculated using this formula. The treatment was applied by adjusting the soil water content to 4 levels field capacity and calculated by this formula. Observation of variables were measured from 1 to 7 weeks after planting. Observation of parameters, plant water status, was determined by calculating the leaf spread mass, its target mass, and its draw mass. Photosynthesis rate was measured by a plant photosynthesis method, and water use efficiency was calculated as the ratio of the net photosynthesis rate to the transpiration rate. Despite drought-reducing leaf numbers, hitam cempo and putih mutiara proved resilient, maintaining high RWC over 80%, which supported better photosynthesis in putih mutiara. Putih Muntik Wangi Karanganyar, Putih Mutiara, and Merah Sengreng showed the highest water efficiency, while Air 64 had the lowest. Local rice varieties like Putih Mutiara and Hitam Jumbo show better drought tolerance than Air 64, making them significant genetic resources for breeding new drought-tolerant cultivars. This is important because the scarcity of such genotypes has slowed the development of drought-tolerant traits. So this evaluation of the agrophysiological responses of rice will contribute to first, development of drought-tolerant rice cultivars, second, addressing the scarcity of the nutrients, and last, basis for further research. That concludes our presentation. Thank you very much for your attention.
For the next presentation session, entitled The Role of Beneficial Soil Microbes in Building Resilient Food Systems and Agriculture, a review. If you have any questions regarding the previous presentation, please kindly write them in the chat box.
Hi everyone, my name is Destidian Amalina and I am affiliated with Department of Soil Science and Veteran Yogyakarta. I will present our review on utilizing biofilm forming soil microbes for climate resilient agriculture under drought and salinity stress. Climate change is causing more drought and increasing soil salinizations, with lower crop production worldwide. Over 20% of farmland globally is affected by salt, and water shortage are getting worse. These problems threaten food security, especially in developing areas. The microbes that form biofilms in soil provide a natural solution. They can enhance plant tolerance to drought and salinity, decrease the need for chemical fertilizer, and support sustainable and also climate-smart agriculture. These tiny allies have the potential to improve farming resilience significantly. Drought and soil salinity are rising around the world. Threatening food security, climate change is making this problem worse. To protect food productions and the planet, we need solutions that support zero hunger and climate actions, and the beneficial microbes are essentials to this effort. This review connects microbial biotechnology with climate resilience. We analyze studies on soil microbes that create biofilms and enhance plant growth under drought and saline stress, identifying key mechanisms that can be applied for sustainable crop improvement. We follow the PRISMA guidelines to conduct a systematic review over 20 papers from 2020 to 2025, focusing on microbial species, their function, type of stress, and plant response, resulting a clear evidence-based summary. Our result shows that biofilm-forming soil microbes serve as growth promoters and microbial shields. They produce hormones and boost antioxidant activity, helping crops endure drought and salinity. On the average, they increase root growth and biomass by 60% and enhance yields by up to 40%. The farmers can use helpful microbes to reduce the chemical fertilizer and pesticide. This lowers carbon emissions and improves soil health and biodiversity. In the long run, it creates a stronger farming system where healthy soil support healthy plants and healthier planet. This work promotes two key sustainable development goals, which are zero hunger and climate actions. By incorporating microbial biotechnology into agriculture, we can enhance food security, restore soil health, and lower the carbon footprint of farming for a sustainable future. So thank you for your attentions. I hope this review inspires more sustainable solutions for agriculture facing the climate stress. If you'd like to discuss collaborations or share insight with me, please feel free to reach out to me by this email.
For the next presentation session, entitled Meta-Analysis of Soil and Microbial Drivers of Greenhouse Gas Emissions in Amended Rice Fields. If you have any questions regarding the previous presentation, please kindly write them in the chat.
Hello everyone, thank you for your time. I'm Putri Aditya from Universitas Pembangunan Nasional Veteran Yogyakarta. Will present my research on a meta-analysis exploring how soil and microbial factors drive methane and nitrous oxide emission in the rice field. Since rice cultivation is a vital for global food security, but is also a major source of methane and nitrous oxide, this emission depends strongly on water, soil, and nutrient management. As rice demand grows, maintaining productivity while minimizing emission become a key sustainability challenge. Recent research show that rice system can be both productive and climate smart, improving irrigation timing, fertilizer balance, and organic amendments can reduce emission. However, we still lack clear understanding of which soil and microbial mechanism drive this change. Since rice is a staple food for half the world population, but still remains largest greenhouse gas source in agricultural sector, enhancing nutrient balance, compost use, and irrigation efficiency can maintain yield, improve soil health, and mitigate environmental impact. This meta-analysis addressed three aims. First, identifying the main drivers of methane and nitrous oxide. Second, assessing the effect of conventional practice. And third, we want to determining the best management option that lower emission without reducing yield. To achieve those goals, we screen field study from Web of Science and Scopus on monoculture rice that reported methane, nitrous oxide, soil properties, and microbial genes in one paper. The data were extracted using a web plot digitizer, standardized and analyzed with RStudio to achieve our goals. We found that AOB abundance is the key predictor for both methane and nitrous oxides. Methane was linked to nitrate, while nitrous oxide was linked to ammonium. Biochar is the best amendment to reduce both gas, with increasing end rates reducing methane but increasing nitrous oxide emission. Combining biochar with an input about 120 to 80 kilograms per hectare produced the lowest methane and stable nitrous oxide levels. This integrated strategy support cleaner, more climate resilient in rice system. We hope that we can contribute to convert fragmented global data into practical on-farm guidance, balance nitrogen and sustainable organic input reduce emission, and we hope we can achieve the global goals in the SDGs number 2, 12, and 13 and bridge scientific synthesis with climate smart farming practices. And thank you. That's all for my presentation. If you have questions, I welcome your questions and look forward to discussing more with all of you
For the next presentation session, entitled Evaluating Drought Index Using NDDI Method on Toposequence of the Western Lau Mountain, Indonesia. If you have any questions regarding the previous presentation, please kindly write them in the chat box.
Dear ladies and gentlemen, let me introduce myself. My name is Akta Feherawati from Department of Soil Science, Faculty of Agriculture, 11 Maret University, Indonesia. I would like to present our research about evaluating drought index using NDDA method on toposequence in Western Lawu. The background of this research is that Indonesia, located in tropical monsoon climate region, making sensitive to drought and differences in elevation, rainfall, soil type, and land use contribute to vulnerability to natural disasters such as drought. There are many methods for analyzing drought index, including the normalized difference drought index method. The NDDI method uses two parameters, namely the normalized difference vegetation index or NDVI and the normalized difference weather index or NDWI. Karanganyar Regency has a tropical climate with an average annual precipitation of 4,000 per year, and they have the differences in elevation and precipitation will impact in water availability, soil temperature, and the intensity of sunlight. The objective of the research are to analyze the drought index using NDDI method on top of one of lava mountain, analyze the pattern of drought index using NDDI method in 5 years, and analyze the effect of rain characteristic on drought index. All the objectives are to encourage the sustainable development goal number 13. The method of the research is descriptive exploratory, first corrected and grouped radiometrically, and then analysis using NDVI and DWI, and then classifying the drug index using 5 classes. The results show that NDVI for 5 years had only values ranging from 0.13, and the highest were observed from June to September. NDDE visualization in 5 years shows a monthly and annual drug variation, especially during the rainy season transition from dry to rainy season and dry season phases. The peak of drug occurs during the dry season July at elevation 200 until 600 meters above sea level, and the ANOVA results showed that land characteristics including elevation, slope, and rainfall had a highly significant effect on the drought index. That's all my presentation. Thank you for your attention. I am really looking forward to your opinion and suggestion to end this research. Thank you and see you in the next ICC.
For the next presentation session, entitled Evaluating Drought Index Using NDDI Method on Toposequence of the Western Lau Mountain, Indonesia. If you have any questions regarding the previous presentation, please kindly write them in the chat box.
Hello, my name is Winnie Prayogi Abdillah. I am from Agrotechnology, Faculty of Agriculture, UPN Veteran Yogyakarta. Today, I am pleased to present my research title, Rainfall Variability and its Challenges for Sustainable Rice Production in Special Region of Yogyakarta. Climate change has altered rainfall patterns across Yogyakarta, making them more irregular and unpredictable. This shift increases the risk of extreme events such as floods and droughts, disrupting rice growth and reducing yields. These challenges threaten the region's agriculture stability and long-term sustainability. Despite these challenges, there are key opportunities. By analyzing long-term rainfall data, we can better understand local climate behavior. This knowledge helps design adaptive strategies such as improved planting schedules and water management to support resilient and sustainable rice production. Understanding rainfall variability is essential because it directly influences planting time, crop development, and yield stability. In Yogyakarta, even small change in rainfall distribution can determine whether a harvest succeeds or fails, affecting both farmers and food security. This study examines spatial and temporal rainfall variation in Yogyakarta and their impact on rice production. By linking long-term rainfall records with agricultural outcomes, we aim to identify adaptive strategies that strengthen sustainable rice farming in the region. I use daily rainfall data from 27 stations and rice production data for 25 years. Rainfall was categorized by intensity, magnitude, variability, and frequency. Trends were analyzed using the Mann-Kendall test and sense-log estimator, followed by correlation and regression analysis with rice production. The results show increasing trends in rainfall intensity, magnitude, and frequency across most stations. However, some stations indicate declining rainfall frequency, suggesting drier conditions. Rainfall variables showed weak correlation with yield, yet intensity and total rainfall remain key factors affecting rice production. This study highlights historical rainfall patterns in Yogyakarta. Rice need adequate water during vegetative and flowering stage, but less during grain filling. Excess rain at the wrong phase reduces yield. Understanding these patterns help improve planting schedules, variety selection, and on-farm water management. This research provides a scientific foundation for understanding rainfall variability and rice production. Its findings can support adaptive agriculture policies, improve irrigation planning, and help farmers strengthen local food production system against climate change. Thank you for your kind attention. This research is still evolving and further development is needed. I welcome discussion and future collaboration through the email address shown on this slide. Thank you.
Thank you so much for your attendance and participation. We will now have a 15-minute break. The next session,